I'm going to be presenting on a medicine known as Tildrakizumab. This is a antibody that specifically binds to the P19 moiety, coming to this picture, excuse me, of the IL-23 cytokine. And I've also, as you see here, there are some gray bars next to P40. There is a medication known as ustekinumab that's an interleukin-12 and 23 inhibitor that binds to the common P40 unit between IL-12 and IL-23. So this medication, Tildrakizumab, is very specific for inhibiting the cytokine interleukin-23. We know that interleukin-23 is highly important in the stimulation of Th17 cells and the whole pathway that follows, including IL-17 inhibitors, IL-22, et cetera. And it's been particularly important in the realm of psoriasis, and now we know psoriatic arthritis as well. There are at least two other, actually more than that, but two that are fairly well advanced interleukin-23 inhibitors besides Tildrakizumab, and those include guselkumab and risankizumab. I'm also making the point as new medicines become available, they become more unpronounceable to us. And so here we go with Tildrakizumab. Oh, I should mention that these are my disclosures. Pictures are allowed, and I've already indicated what we're doing here. And we're looking at the 24-week data for treatment of psoriatic arthritis. Our primary efficacy endpoints are going to be the ACR20 response, which has been the standard key outcome measure in psoriatic arthritis trials. As you know, this is a measure that's been borrowed from rheumatoid arthritis. There are some newer measures that are being developed that are specific for psoriatic arthritis, but still the ACR20 remains the primary endpoint. And then I'm also presenting ACR50, 70 data, skin responses as measured by PASI 75 and 90, the patient pain, as well as an enthesitis index. And then I'll be covering some of the safety data. So the inclusion criteria are noted here. These are all patients that have criteria for psoriatic arthritis. They could have been on stable background nonsteroidals. They could have been on background methotrexate or leflunomide or prednisone in low dose. They also could have been on at least one biologic treatment, which would have been a TNF inhibitor. This was restricted to 30% total of the population. The key exclusion was any prior use of a medication that was in the IL-17, IL-23 pathway, such as one of the IL-17 or IL-12/23 inhibitors. Study design is as noted. There were several different dose arms of Tildrakizumab, 200 milligrams administered every 4 weeks subcutaneously, 200 milligrams every 12 weeks, 100 milligrams every 12 weeks, and then a dose that was intended to be lower than an effective dose, which was 20 milligrams every 12 weeks, and then there was also a placebo arm. I should mention that this drug is approved for the treatment of psoriasis in multiple parts of the world. In the US and Australia, the standard dose is to receive Tildrakizumab 100 milligrams at baseline, then at 4 weeks, and then every 12 weeks thereafter. In Europe, the 200 milligram dose is also available, but in general, clinicians start with the 100 milligram dose. As mentioned, the patients could not, there could have been no more than 30% of patients that had been treated with a previous TNF inhibitor. There was stratification based on weight, more or less than 90 kilograms. Background oral DMARDs could have been used, such as methotrexate or leflunomide. There was also the possibility of an escape if the patient had less than 10% improvement of tender or swollen joint count at 16 weeks. As it turned out, no one escaped. So the patients, all of the patients in the trial remained on their original dosing regimen through 24 weeks. These are the baseline disease characteristics, and the most important point is that these were relatively well balanced between the different treatment arms and typical for a psoriatic arthritis population, including being about 50 years of age, being equigender, being having psoriatic arthritis duration between 6 and 7 years, and slightly less than 20% of the patient population had been on a previous TNF inhibitor. Baseline disease severity was also well balanced between the treatment arms. As you can see, there was active disease with a number of different swollen and tender joints, elevations of patient and physician global assessment of disease activity. About 2/3 of the patients had evidence of enthesitis, and using an enthesitis score system known as the Leeds Enthesitis Index, there were typically around 3 tender enthesial sites for the patients that had enthesitis. You can see the amount of psoriasis that was involved as well, and a function questionnaire. This is the primary endpoint, the ACR20, and as you can see, all 4 dose arms of Tildrakizumab achieved statistically significant separation from placebo at the 24-week mark, with 79.5% in the 200 milligram Q4 arm achieving this, 77% in the Q12 arm of 200 milligrams, 73% in the 100 milligram. And even with the 20 milligram 50, I'm sorry that the graph doesn't allow us to read that, but these are all separated from placebo. Interestingly, the placebo response was 50% at the 24-week mark, showing a climb from the week 16 mark. But it's mentioned, although you might have thought that the possibility of an escape could have somehow influenced this, no patients did escape, so that was not an explanation for it. And so we don't really have an explanation for why the high placebo response rate. The effect size, that is the difference between the placebo and Tildrakizumab, was similar to what we've seen, or slightly better than some of the other interleukin-23 agents. This is an issue that we do see in clinical trials, that sometimes there is a high placebo response rate, so we're depending upon a very high absolute rate in order to see of the treatment arms in order to see a separation. Here we're seeing ACR 50 and 70 responses, and I would comment that the ACR 70 is a little bit more realistic in terms of the placebo response, just 10%, and and clearly separated from the majority of the Tildrakizumab arms. Moving over to skin response, this shows us that there is a high degree of effect in the skin, what's known as a PASI 75 response, which is a composite score of the skin, including erythema, induration, and scale, as well as a body surface area ingredient. This was shown in approximately 80% of the patients who were in the 200 milligram every 12 arm, and 64% of those in the 200 milligram every 4-week arm. This is a more objective measure, if you will, because you're just looking at the skin for the scoring, and so we see a lesser degree of placebo response here. PASI 90, which is a higher threshold for skin response, 50% achieved this in the 200 milligram Q12 arm. And all of the dose arms were clearly separated from placebo. Now we're looking at the pain change, and what we're showing here is that all of the different Tildrakizumab arms were statistically separated from placebo. There is a relatively high placebo response rate, though. There is about a greater than 50% improvement in pain, and most patients would based on the pain literature research, this would be considered a major clinical change. Enthesitis, this is based on pressing on typical ligament or tendon insertion sites in various parts of the body, which can be involved with inflammation and pain. And I will say that sometimes in clinical trials we tend to see a high placebo response in this measure, and this was also noted here. And always in the mean change, we just saw a difference between the 200 milligram Q4 arm and placebo at the 24-week mark. There are further analyses underway as we speak. This is very fresh data. And so, for example, we're going to be doing an analysis of what we call complete resolution, just to see if that has any better way of distinguishing from placebo. If we turn to safety, here are the frequency of treatment-emergent adverse events, serious treatment adverse events, which were relatively low, and there were no due to treatment-emergent adverse events. The one serious infection was a case of tonsillitis, and there were no major adverse cardiac events, malignancies, although this was a relatively short trial, or deaths. And there were no significant changes in various laboratory parameters. So to conclude, by week 24, all 4 doses of Tildrakizumab were significantly more efficacious than placebo in treatment of both the joint and skin manifestations as well as pain. There was a clear separation between the Tildrakizumab arm and placebo as early as 8 weeks. And it appeared that shortening the dose interval from 12 weeks to 4 weeks really didn't make much of a difference. What we've been observing with all of the IL-23 agents is they tend to have a prolonged efficacy. We especially have seen this in the skin of the psoriasis clinical trials. And so it appears that Q12 dosing is going to be the appropriate way forward. And relatively little in the way of safety signals were seen. So with that, I will open up for any questions.